Hello, everybody. I'm Martin Steinegger, an assistant professor at the Seoul National University in South Korea. And today I'm going to talk about metagenomics tools that we have developed. But first of all, I want to thank the organizers for giving me the chance to talk at this conference and to have the chance to explain our research to you. So what you can see here in the first slide already are these like cute little fellas in the bottom, and each of them represents actually a method. And today we're gonna talk about some of them, and we're gonna talk about this first one on the rocket, which is a search method for homology detection. And we will talk about protein clustering. We will talk about assembly, and we will also go a bit about a web server we have built and where you interactively can explore your data, and maybe talk a bit about contamination in your data. These pieces here on the right is actually also quite interesting software. It might be for you, might be interesting software for you. And one is for eukaryotic metagenomic assembly, and the other one is Spacefarer, which is for phage detection. Each of these tools, has some publication behind it. Please feel free to check any of them out if you're interested. But yeah, as mentioned before, we will be mainly focusing on search, clustering, and assembly and contamination. For this tutorial, we are using Conda, which is a package manager for software. Especially we use BioConda, which the biological community use to share their software. So there are two commands here. One is the Conda create, which will generate an virtual environment for you called tutorial, and it installs for you MMseqs2 and PLASS. And the second cell here just activates this tutorial, this virtual environment, and it sets up the path for you, so MMseqs is ready to be used. I have already done that, so this is then the expected output that you should see. I don't run the first cell here because it takes a bit of time to solve the environment and to install the software, so I already have pre-installed it. Today, we're going to talk about a case where a 60-year-old man came to the hospital, a hospital in Boston. So first of all, this is not data that we have produced, it's public data, but I'm just using it to example how to use our software to solve this case. So this 60-year-old patient has some meningitis, and the doctors looked at him, and what they could see is that there were like some tick marks. They could find some tick bites. He was actually, he's a woodworker, so there might be some exposure to rodents or like some bats as well. And it seems like over the time, the patient is getting weaker and weaker, and the symptoms getting stronger and stronger, and so the encephalitis is getting better. So the doctors try to find a way how to diagnose this case very fast and come up with a new way using NGS data. So in this tutorial, our task is now to use the data that they produced to figure out what is the disease causing the meningitis. And on this slide here, you can see a comparison of pathogen detection methods currently used in hospitals on the left side. It's a slide from Simmons from Johns Hopkins University. You can see based on the pathogen that you might have, you have to run all kinds of different assays in the hospital, and some of them take some days. But like for example, fungi or so, it can take weeks until you get a diagnosis back for your disease. And this is really critical time, right? So because the patient has, for example, now this meningitis, and we don't have weeks maybe until we can get an answer. So here on the right side, you can see a workflow that is based on next generation sequencing, is where you take a clinical specimen, best from the infectious site. You extract the DNA, and then you run some NGS on it, so you sequence it, and you get reads, and then you start to try to classify that reads and maybe figure out what might causing this disease. And the turnover time for this is only a few days, so from maybe up to 2 days. Here on the right side, what you can see is actually an Oxford nanopore sequencer, and using this Oxford nanopore sequencer, probably the time of the sequencing can be reduced rapidly, so we probably can have diagnosis within hours using nanopore. So I assume that the future will be somewhere where we are combining still old tests, but we will transition slowly to NGS tests, because they're just having better turnaround times. And also, one other thing is NGS does not need to distinguish between these different types of parasites you're looking at. It's just one test to rule them all in general. How you start with this NGS pathogen detection pipelines is you start with some organism you want to look at. Here we have human. You sample. So here we took a sample from the spinal fluid, and then you do DNA extractions. And already here some things can go wrong. So you can do DNA and RNA, but to do DNA extraction, what you have to do, you have to break the cell walls of the organisms. And some cell walls are thicker than other cell walls and harder to break. So you might need to use different methods to break them, for example, like beading or so on. And once you have your DNA or RNA, you put it into a sequencing machine, and then normally millions of short reads fall out that are having the length of 100 to 250. And you take these short reads, then use some software and search these short reads against reference genomes or reference databases. And then you can answer the question like, who is there? And who is there is in general, you try to assign each read here a taxonomical label from the reference database. And based on this labels, you then can decide who is there and what might cause the infection. So for this case here, they sequenced the brain. They performed a biopsy at day 8. But they also did the CSF before. And it returns 14 million reads. And the authors only provided the reads that where no human was matched for like privacy reasons. So they initially took this 14 million reads and they used the tool Kraken to remove all human reads from it. Yeah, so the question is, again, why would you not release everything? But as mentioned, privacy is very important, so we cannot just give out human DNA. To explain what Kraken does, because Kraken is really one of the standard tools to labeling these reads, is so Kraken takes a read. Here on top, you can see this read, and it breaks this read into k-mers. So k-mers, you can imagine, are like short words of a fixed length. So for certain, so I think Kraken uses something like 31 mer or so in default, or 30 mers, I think. So meaning there are 30 residues. So it breaks it into all overlapping 30 mers. And then it looks up each of these 30 mer in a reference database, which was pre-computed already from reference genomes. And whenever it finds an 30 mer match, it takes over the taxonomical label from the database and just colors that k-mer. And once it has computed all of the k-mers, so it found all of the taxonomical labels it can find, it places them into a taxonomical tree. So here you can see you have some orange, blue, and red, and so on. And it then decides that the label of the query sequence will be the one that has the most support, like the heaviest pass in the taxonomical tree. So it will give the label to the query sequence. So there's not much more to it. In generally, a k-mer lookup followed by a weighted tree search to give taxonomical labels. So the question is now, what are the limitations of nucleotide-based classification? One problem with nucleotide classification is that we can only find hits in the database that are close to the reference. Everything that diverges a bit further away is hard to match just using nucleotide information. But there's a trick we can use in order to sometimes even find these remote homologous detections. for this, we can match things on the protein level. So for this tutorial, we're going to talk about MMseqs. MMseqs works on the protein level, so we will do that. But keep in mind, the reason for doing so is if you have a sample and there are things in these samples that might be hard to match to some reference, then you might want to use protein searches. And the reason is why do you want to do so? Because proteins are longer conserved, like over a longer evolutionary time. What you can see here actually are structures for ubiquitin in human and ubiquitin in yeast. And by just looking in this three-dimensional structures, you can see that they look very similar, right? So they are still super similar, even though there's billions of years of evolutionary time in between them. Reason for that is they have an ancestral protein structure, which we don't know anymore. And if you look at the sequence over time diverges. So you see that only by looking at the colors meaning there are different biochemical properties, that these sequences have changed over time, but structure stays the same. But some patterns in the sequence are actually also still conserved. So you have this red block here, for example. And these protein search methods actually have the possibility to pick up even diverged signals like that. So you can still find connections that are really far away. So in order to make this search, we'll use MMseqs2, and we will align these reads against the protein databases. The question is now, how can you really do that? So how can you take a DNA read and align it against a protein database? One trick you can do is called 6-frame translation. And 6-frame translation works in a way that you look at all possible reading frames. So there are 3 possible reading frames on the forward strand, and there are 3 possible reading frames on the reverse strand. So you can just make all the codon combinations, like just start here, take your codons and so on. And then you will have approximately 6 times the amount of sequences. But you can search them then against the databases. Now you can say, okay, but most of these frames are useless. It's true, but most of the frames will also not match any protein in the database. So only the frame that really encodes something curative will be matched, most likely. So MMseqs consists of multiple stages. The reason why we need multiple stages is because we have to be really fast. In metagenomics, we work normally with really big data sets. So a sample can have gigabytes or like hundreds of gigabytes. So we need to be able to match these DNA reads as fast as possible to proteins. And to do so, we first use a fast prefilter based on k-mers. So we have learned what k-mers are in Kraken. So in Kraken, we have that 30 mers. Here are 7 mers, but they are protein on the protein level. What MMseqs does, it tries to find 2 7 mers on one diagonal. Once it has found matches where it finds these 2 hits, it makes an ungapped alignment of this diagonal. And if the diagonal is above a certain score threshold, it makes a real Swiss Waterman alignment. Swiss Waterman alignments are expensive to compute, so you try to reduce that as much as possible. Finding these k-mer hits can be really fast, and ungapped alignment is also relatively fast, but you really want to reduce the amount of real alignments you have to compute at the end. Important for these prefilters is that they should be sensitive as well, like not just fast. If you lose a lot of sensitivity, then your prefilter is also not really useful anymore. So with this combination, we found something that works good for us. And we made a comparison of MMseqs against other search methods. Here, these red bars are MMseqs. MMseqs can have different trade-offs. It can be very fast and not very sensitive, what you can see here, or it can be very sensitive but not as fast. Just to mention here, on the x-axis is the sensitivity, and on the y-axis is the speed up relative to BLAST, meaning the best point you can have is actually somewhere in the upper right corner, I guess somewhere infinite, hopefully on the speed up. But we see that these are sequence search methods, and none of these fast methods actually reach the sensitivity of BLAST, or like only MMseqs gets in the range of it. Here's a question for you. The question is, you have an alignment. So here you have an alignment between two sequences, and this alignment has an e-value of 0.2. Do you believe that this alignment is homologous? Do you think this is a real, genuine hit? So the question can be, normally if people ask this kind of question, the answer is always yes, I think. But here what you have to keep in mind is that an e-value of 0.2 can be fairly random. So you have quite high chance that you actually by randomly see an alignment that looks like this. So I guess if you really work with that and you want to publish something, I guess you would not trust this alignment. But if you, instead of aligning two sequences, align a multiple sequence alignment against the sequence, you can find conserved patterns over the history of time, and much better. So here you have this like really well-conserved red columns, really well-conserved green columns, and you see that they line up pretty well between the query and the target. And by doing so, you can establish that the e-value is actually much, much better. So you can find things that are much further away than if you would just compare two sequences. And this is what PSI-BLAST introduced. And it does really give a big boost to bioinformatics. So we're actually now able to find things that are really, really far away by just using these profile information. And MMseqs is also offering these kind of searches. So you can do these really, really diverse searches, profile searches here. You see that MMseqs, these small numbers indicate iterations. PSI-BLAST can search iteratively, where it like picks up and hits on the way, like makes a search, finds hits, refine the profile, search again, and so on. So it's 2, 3, and 4 iterations. You can see that MMseqs is more sensitive actually than PSI-BLAST here, and much, much faster. So it's also possible to use now profiles at a metagenomic scale, which really helps for like finding hits that are like hard to find. And now we'll guide you a bit through MMseqs. So MMseqs has this syntax that is a bit similar to Git. Maybe if you have worked with Git before, you have your MMseqs binary, and MMseqs comes with commands. So MMseqs normally you call a command, and then it defines input databases and output databases, and then you can have your flags afterwards. Let's just look at it when we use MMseqs and see what the binary does. So if you click on it, you already see that by just calling that MMseqs binary, you see that it has like multiple workflows. We have a difference between easy workflows or like more involved workflows. Easy workflows are really workflows where you take a faster file input, make a faster input, and then you get your output out. Like it's just a single command that runs everything for you. While MMseqs is also built to be a bit like a database system, I guess. It was not initially planned like this, but it ended up being one. The idea is that you have a lot of modules, I think hundreds of modules, where you can do searches, and then once you have done a search, you can filter it, and then once you have filtered it, you can compute the LCA of the hits, and then if you like that, you turn it into some taxonomical report and so on. So MMseqs offers a lot of the possibility to plug in multiple search options or like filter options. But if you want to do that, then that becomes a bit more involved, and you probably need to read more of the user guide. But today we're going to explain a bit how to use these modules actually. So first what we have to do, we have to download the reads. So these are the reads from the patient. We have already pre-processed them in a way that we use the software Flash. What Flash does, it takes paired end reads from a sequencer. So you maybe might remember you have a forward read and reverse read. And if these reads have an overlap that is big enough between them, then Flash just merges them into a single read. If not, it just keeps the read. So these mystery reads are already processed by this software. It helps you to increase a bit the length of the read sometimes. We use reference databases. MMseqs has actually already a module called databases, and databases helps you to set up databases so you don't really need to download the FASTA file from the NCBI or from UniProt. You can use MMseqs databases, and it provides, for example, UniRef, the UniProt, the NR, the NT, PDB databases, structural databases, all kinds of profile databases, and yeah, the list goes on and on. And you can just easily just say MMseqs, give the database name, then the output sequence name, and the temp directory, and then it will download the data for you. It will set up the database, and you're good to go to search against that database. For this example here, for our tutorial now, we're not going to do that because we wanted to keep the database really stable. So we wanted to have a fixed version so that all of the results that we are reproducing here are as we want to see them. For this tutorial, we are using an old version of UniProt, and actually the Swiss prot of UniProt, from the year 2018, just to keep it stable so that all the results are reproducible. So with the first commands, you're just downloading the old Swiss prot. The second one is a mapping from the Swiss prot identifier to a taxonomical identifier, and then you just unzip that. I have already done that because I'm sitting here in Korea. Somehow the download takes some time. I guess it could be much faster if you're closer to Germany, because it's currently hosted in Germany. Once you have done that, you create an MMseqs database by giving that FASTA file to it, and you define an output database. So let's just execute that. It should run and tell you that it converted some sequences. Now it's done. Now we're turning that sequence database into a taxonomical sequence database. And if you just call createDB, you can call on a single FASTA file, but then you don't really know what is the mapping from a FASTA entry to a taxonomical entry. So with createTextDB, you can now assign taxons to each of these entries in the database. Let's make that small here. And what that does is actually it downloads in the background the NCBI taxonomy. In default, MMseqs uses the NCBI taxonomy, and that takes a bit of time. But once this is done, we have a set it up database. Okay, here also my download speed is not very fast, but we are at 100% and the database is created. And as a next step, we create an index. So indexing means that you take the sequences and you put them into a shape that MMseqs fastly can search through that. I have mentioned these k-mers before. This create index step actually extracts these k-mers and stores them then on disk afterwards. So this is also done already. So indexing can take a bit of time if you have really big databases, but you only have to do it once. You can then search against the database multiple times if you want to. Okay. We have our mystery reads FASTA file, which we have downloaded before. We can also turn that into an MMseqs database, same way. So this is done. It tells you its type of nucleotide, so it infers the type of the sequences automatically. So you don't really have to write if it's like a protein or a nucleotide. And then what we can do is we can call a taxonomical workflow. This taxonomical workflow will take these reads now. It will search them against the UniProt sprot database we have set up, actually the index that we have generated. And it will generate lowest common ancestor results. Like it will assign actually each read a taxonomical label. We will explain how it does that in the following slides. And we have defined one parameter, -S2, is reducing the sensitivity of MMseqs just to be faster. For this example here, we don't really need to be as sensitive as possible. But maybe for your use case, you might want to adjust that. And this might take a bit of time, but MMseqs always tries to tell you a lot. Some people like it, some people hate it. The thing is, it tells you each step it does. So what it does first, it says here, oh, I'm extracting ORFs. So it takes the reads and extracts ORFs, meaning this is the 6-frame translation in general. Then it searches these ORFs with a prefilter against that index. It finds hits. It then performs an alignment. So it does the gapped alignment. And then it does some logic with offset alignment, meaning since it was nucleotide before and then protein, we search on the protein level, we have to offset it back to the nucleotide coordinate system. There's some filtering going on. And then after the filtering, we compute the LCA. So what I meant is MMseqs has many of these modules. What you see here is that this workflow of taxonomy is nothing more than a concatenation of multiple MMseqs modules. So if you think about it in your case, right, you might not want to use exactly that use case. You can just change that workflow freely and like adjust filters or add other things that you want to build your own method for taxonomic classification. And so MMseqs uses this 2B-LCA protocol, which was comes actually from the marine metagenomic community, where the idea is that you want to consider uncertainty when you give a label, taxonomical label to a sequence. In the nucleotide level, right, if you match something, it's very likely that this thing is really the thing you have matched, because it's just hard to match really diverged hit on a nucleotide level. But on the protein level, you have a lot of possibilities to find things that are really far away. So the 2B-LCA workflow, what it does is it searches against the database, it finds a best hit. Once it has found it, it has an alignment start and end position, it cuts out that best hit. And it also recognizes the E-value that it had for finding that best hit. Now it searches actually the cut it out sequence against the same database again. It finds multiple hits now, and it accepts all hits that have an E-value that is better than the best hit from before. And then it places them into a tree, and it gives the label for that. Why you do that, right? This kind of measure considers uncertainty. So the better your initial hit is, your best hit is, the less hits you will find in your second search. And therefore your label will be closer to species. But if you found something really far away in your first search, then you will find way more hits in the second, and then your LCA label will be higher up in that tree. So it will adjust for uncertainty. Once you have run that, MMseqs, we have stored the LCA result in a file called LCA result, and we can convert that now into an TSV file by calling the MMseqs create TSV command. So you need to just define the read input database and the LCA result database, and then you get your output LCA TSV. And let's have a look what that looks like if you really run that. So what it does here is that the head of that, just the first 1,000 lines, right? It tells you this is the mystery read identifier. This here is the numerical taxonomical identifier, and then you have the species name behind it. So it assigns reads to taxa. You see many of these are still unclassified. Could be one reason because we didn't really increase the sensitivity a lot, but also maybe some of the things we really don't know, or some of them might be technical biases in the data. So you can see there's some bacteria. There's all kinds of bacteria here, but many things are unclassified. So you can just use this command here, for example, to find out what are the taxas with the highest read counts. And you will see that there is actually, the biggest one is unclassified, and still a lot of them are unclassified, but the one that we can classify are some bacteria. And then the third one is some tick-borne virus and E. coli. We also still have some Homo sapiens in it, actually. I have mentioned in the beginning that we filtered, or that the authors filtered this data set before for human, but it's really difficult to get rid of all humans in the sample. There will be always something left, and especially now we're matching on the protein level, we are more sensitive, so we can find them, meaning that, yeah, Kraken might have been not sensitive enough to annotate these few human reads. So the question now is, does it make sense that there's so much bacteria in that sample? It's a clinical specimen. It's not an environmental data set, right? We would not expect that there's actually so much bacteria reads here, at least when I worked first with pathogens, I did not think this was the case. But yeah, we have many problems with this. It's like you have lab contamination, you have all kinds of sources of contamination that goes into that process. So you might pick up some signals that are actually not real signals. So probably most of these bacterial hits that we have here are not real. I think I have Wikipedia'd this species before. And this is actually a plant pathogen. So it's very unlikely that this is probably in humans. And so it's most likely contamination. So the question is now, how do we actually determine what is the pathogen or what is not a pathogen in this case? And okay, first thing we can do is now we can summarize these LCA results into a single report. That might be easier to look at than what we had before. So these LCA reports are, if you have worked with Kraken, are in the Kraken format. This is just like a tree format of all the LCA labels that you have, that we have generated. We can just get that. And so now what that visualization here is doing, it tries to put in taxonomical tree over that data. So it tells you that, okay, 93% is unclassified, then you have cellular organisms, you have bacteria and so on. And we have these bacterial hits we have found before. Now the ones that are close to each other are also close in this file. And yeah, but just scrolling through that, there are many hits that do look very unclear, right? So we have so many possible candidates that can cause this infection. So what we normally would do now in these cases is to use a software called Pavian to load this report. We will do it in a second. But one thing to keep in mind, normally if you do these experiments, you have samples from the infectious site, but you also have a sample from a control. So you sequence something like water or something in your lab. So, and then you subtract everything that is in the control from this report, because if it's in the control, then it's probably just lab contamination. And by doing so, you're getting rid a lot of the you're getting rid on a lot of these signals here actually. However, for this study, the authors did not provide us a negative control, so we have to work with what we have at the moment. So as a next step, we are loading these reports into Pavian. So it's the visualization tool I have mentioned. So Pavian offers a Shiny app, meaning that you can just actually, if you want to, you can just copy paste that URL. You can put that into your browser, and it will give you, once you push enter, it will bring you to a site that looks like this. In the beginning, you have a selector, so you select some reports you want to look at. Pavian works with multiple software. It works actually with Kraken, Centrifuge, Metaflan, like a lot of software for taxonomic classification. So you can push browse, and then here we are in the folder of that current notebook. I'm giving it the report.txt, which is that Kraken, like this is this tree that we have seen just as a file before, but we can now load that here into that visualization. And if you do so, you can click on sample, and sample will give you that tree that we have seen now as a file before, but now in a visual manner. And you can see that 20,600 reads are bacterial, and then out of this 18,100 are from protobacteria and so on. So you can see how the reads distribute over the taxonomical tree. You can see that there are some eukaryotes here in that one, which is, I guess, yeast. Then you have some viruses here. The biggest virus is the Powassan virus. One other feature you can use, which we currently do not, I mentioned that you would like to check against a negative control. So you can click on comparison. Comparison now gives you a table of these species. If you load into the tool multiple reports, you actually can see the read counts lined up next to each other from the multiple reports. So you could have your negative control and your positive control. a positive sample, and then you can just see what are the reads differences for these taxas, and you can filter the ones that are only in the positive one but not in the negative one, and that really helps you to narrow down what might be causing this infection. Again, these tools, right? These tools can be used not just for infection detection, but you can use it for all kinds of metagenomic tasks. It's a really handy tool to look at data. If you don't want to use Pavian for some reasons, there's a way that MMseqs can report corona reports for you. Let's just do that. So you can, once you have your taxonomical output, right, we're using this LCA result file from before that we have computed initially, where we assigned each read in LCA. And we can just say, make a taxonomical report. We give the report mode 1, which is for the corona reports, push that. It has to load the NCBI taxonomy and so on. But then if this is done, then we should have a ready report. And we can visualize that report. So that report is an HTML file. So it means if you're working on your workstation, you can just double click on it. If not, here we currently embed that as an iframe. And we can see now that here the cellular organisms, we can click on them. And yeah, we see that the biggest fractions are the bacteria and so on. And we can find our viruses somewhere, I guess. Do we? Here. Oops, these are the bacteria. Yeah. Anyway, it should be the same data that you have that you also have in the Pavian visualization, but it's just a different way to look at it. Some people really like these chroma plots to look at their data. Okay. Just to mention it, right? We have done like multiple manual steps now to get these reports, and maybe that is a bit tiring for you and you don't really want to do that. So you can use the easy taxonomy workflow that I've mentioned before. These easy taxonomy workflows are where you give in reads and then you give a database, and it should generate most of these reports for you. It should generate the Kraken report for you. It will generate the TSV file for you automatically. I think the corona we automatically generate. So you just call that one command and just generates all of these things. But then once you have done that, you cannot stay in the MMseqs framework anymore. So you cannot use the downstream modules of MMseqs. That's the disadvantage of it. But if you're only interested in the labels, then this is an easy way to go. But let's just call that as well, because the easy taxonomy workflow actually generates one report for you that it's a bit hard to report if you just use the normal MMseqs modules. So you need to call multiple modules to generate that. And it's a top hit report, which can be quite valuable if you especially look at pathogens. So currently now it's prefiltering the data. It's aligning the reads back. It does exactly the same steps as before, but at the end it just generates also these other reports. So there will be some more output here, like after the LCA. Before we stopped at the LCA, and now we do not stop at the LCA. We do that create TSV to make the LCA report and call the taxonomical report to generate this. So you see, with this like modules, you can compose like all of these more complex workflows. Okay, and now it's done. And now let's have a look at one of these pathogens, which is the Povozan virus. And in this top hit report, what you can see is that there is one protein of the Povozan virus, which is this identifier here, the Q04538, and 1,244 reads mapped to it actually, and 73.8% of that genome of that protein are covered with reads, and the average sequence identity of the hits to that entry is 92% roughly. So it's a bit diverged already from like our reference Povozan virus. It's not really directly that Povozan virus. Overall, that's then 12x coverage, meaning that there are roughly 12 times reads in that unit coverage here. Actually, no, in the length of that protein sequence. So normally, if you look at why is that useful? Like, why is this report useful for pathogens? There's a lot of technical noise that you have from this sequencing experiments sometimes. So what you really want to find, if you want to find a pathogen, you want to find a pathogen that has nice coverage, right? So you don't want just reads that match only to one point of a sequence, because that's mostly a technical fragment. You want something that is like an even coverage over the whole genome to really verify that this is a real thing and not just noise. So if you look at that, that's a bit weird, right? So normally multiple viruses have multiple proteins, or like not just viruses, bacteria also have multiple proteins, right? So you would expect that you see multiple protein hits, but however, for the Powassan virus, we only hit one protein, which is not super convincing, but it's a tick-borne virus, right? And somehow this worker had some tick bites, somehow that seems to be tempting to believe that that might be this virus, right? So when you actually look up that protein in the UniProt, on the UniProt side, you see that this is one of these polyproteins that encodes multiple proteins inside. So the Powassan virus has like one big open reading frame, and that open reading frame encodes multiple proteins. So meaning that in that report before, we have only seen one hit to one protein, but that means it hits, that the reads are hitting multiple proteins at the same time. So it's meaning that it's getting more convincing that the tick-borne Powassan virus is actually something that is in that sample. So now we want to have a closer look at these reads. So normally what you do is, once you establish that something might be the pathogen, you want to follow up on the reads. You want to really get that reads out from that virus and see if you can assemble it, or if you can match it to other databases and so on. If this is really something that you think that looks genuine, then you just really follow up this way. So what we do here is now we just grab Powassan in the lca.tsv file, and we see that the Powassan identifier, NCBI numerical identifier for it is this number here, 39,008. Okay, let's first extract that Powassan virus sequence from our target database, meaning we want to now extract the SwissProt entry. And for this, we use the MMseqs filter taxdb command. We use the taxon ID that we have extracted from the report before, from this TSV file before. And now we turn that into a TSV file as well. And using that, we now subset the reads to actually extract only the reads that are pathogens. And means now in these reads pathogens, we will have our pathogenic reads, meaning these are all reads that are classified as Powassan. And we convert that now to a FASTA file because most other bioinformatics software uses FASTA files as input. And let's check how many reads do we have in that file. We can just count with the grep command how often do we see the FASTA headers, and this is the 1,233 reads that are labeled as Powassan. So as a next step, as I mentioned, is like one thing you could do is you can assemble these reads now, like try to figure out how well does these pathogen actually assemble. For this, we're gonna use in this example here PLASS, which is a protein level assembler. And we actually have built that to assemble really diverse metagenomes, especially from like soil or so on. But I just want to use case it now here and show you that you can also use it for assembling something that is maybe not as complex and not so difficult. Other assemblers you could use were like Megahit or Metaspades. You could just use these reads and give it to one of these assemblers, and they will also do a good job now in assembling them. PLASS was designed, as I mentioned, to really find these, assemble these diverse environments. And why can it do that? It does it because it, instead of assembling nucleotide reads, it assembles reads on the protein level. So it takes the nucleotide reads, it uses again the 6' translation. It finds overlaps between these reads on the protein level. and then concatenates these reads together into proteins, and it does that like iteratively until it reaches the stop codon and the start codon, and it has assembled that. Why does it really work is if you look at this plot here, you can see in the middle there's an overlap between these multiple reads, and because of the wobbly position, right? The wobbly position, the third codon position can change quite a lot. If you just look at the DNA version of it, you can see there are like multiple mutations happen. And once you look at on the overlaps on the protein level, you collapse all of these SNPs. So you effectively get rid of the wobbly position and maybe sometimes also the second codon. So it helps you to find overlaps better. And we have actually for PLASS, we have benchmarked PLASS against Metaspades and megahit in like multiple environments, and wanted to see if we can get more protein information out of these environments. So what we did is we took samples from human gut, some marine eukaryotic set, it's actually transcriptomics, metatranscriptomics, soil, and like a multiple set of soil. And we gave these sets to the assembler, our assembler, which outputs proteins, or like megahit and metaspades, which outputs you contigs. For the contigs, we then call protigal on them to extract the protein sequences from them. And then we compare how much more proteins we can extract at clustered levels. So we're clustering these sequences to remove redundancies from the proteins at 90, 80, 70, 60, and 50% sequence identity. And we can see that PLASS can, in all environments, assemble more proteins from these environments. For human gut, the ratio is like somewhere in between 1 and 2. But for really diverse sets like soil or like multiple set soils, the ratio can be much better. So you can get way more protein sequences out of these environments and use more of your sequence information. So Annika Seidel, she's a PhD student in the Max Planck in Göttingen, is currently working with Johannes Söding and me on an updated version of the PLASS assembler, which should be able to now assemble not just proteins, but really extract contigs out of these environments, but again, using these protein overlaps from before. So what it does is very similar before. You still do your 6' translation, you find your overlaps on the protein level, and then you have you assemble an ORF, but you know, when you assemble that ORF, you have now also a nucleotide sequence on top of that. So it just memorize what was the nucleotide sequence of the reads that you have used. And once you have assembled your ORFs, you do a second step, and that second step is just trying to link these ORFs together using like DNA linking reads. These linking reads, you can imagine, are just like the intergenic regions. And for most, or like for many microbial species, these intergenic regions are actually not really long. So for example, viruses are highly dense and they have often like really overlapping ORFs. So meaning that often the reads can be, one read could be enough to gap these assembled ORFs. And we tested it also on some data. These are some data from a sludge sample from single-stranded RNA viruses. So these single-stranded RNA viruses have always 3 proteins that are crucial to them. This is MPCP and RDRP protein. And in the study that was performed, that was published in 2020 in Science, they assembled and then they looked at the ratio of the core genes that they assembled. And we assembled the same sets with our protein guided assembler and also looked at the ratio of what we get out. And we can see that we capture most of these core genes in a similar fraction. I'm sorry, the colors are really not well picked here. It should match up with the same colors here. For publication, we will change that. And then we also used other assemblers like megahit, metaspades, and RNA spades to assemble these same sludge samples. And we compared the overlap of the context that we have assembled with our PLASS-HYBRID assembler to the other assemblers, and we can see that we can cover most of the assemblies of these other assemblers, but we can also get some more sequences out of these environments. And these viruses and the sequences that we get out are again redundancy reduced, and so we get rid of everything that is redundant. And we check that they have the 3 core genes, so they might be valid single-stranded RNA viruses. So now we will use the assembler that I have just introduced to assemble these pathogenic reads that we have extracted. To this, for this again, PLASS is also similarly structured to MMseqs. It has a module system, so you can call PLASS as well initially, and then it will have like a nucleotide assembler and protein assembler, and then it has this hybrid assemble. And this hybrid assembler is the one I explained where you first assemble these ORFs and then you stitch them together afterwards. We're giving 2 parameters here, like saying how much sequence identity do we allow in the overlaps, which is in general 95% identity in the nucleotide overlap. And then some criteria for like a minimum contact length. So we only want to see contacts that are larger than 200 and so on. And like the minimal ORF length, like for each read, how short should an ORF be that we consider? So let's call that. These assembler PLASS is actually also built upon the MMseqs2 code base. So it reuses quite a lot of the modules that were already developed within that framework. And so the output looks very similar. And again, you can see that it calls first create DB. That's a command that we have learned before. And extract ORF we have seen before. Concat DBs we have not yet seen, but it just in generally leverages a lot of the code base before. It produces quite a lot of output. We have to like narrow that down. And also in this Jupyter notebook, somehow the progress bar doesn't really work properly, so it really just prints it as each line and so on. But here, based on this number, I can see that it actually finished. And so we can have a look at that now. So these are the assembled sequences that it produced. You see they are fairly short. None of them is like particularly long. So optimally, what you would like to see is, since this is this Povosan virus and it has only 1 ORF, you would like to see only 1 large protein that is assembled end to end. That would be nice. But since this is like really fragmented data, it is not that great. So the assembly quality is not that great. So we can ask now the question, how many of these sequences, how many do we assemble? And there are 46 sequences that are assembled. However, the way how our assembly program works is it doesn't care about redundancy. It just assembles things that are also redundant, meaning some of these contacts that we have seen here contain redundancy, might be even identical to another contact. So in order to dereplicate this assembly, we can use a method. It's also part of the MMseqs packages. It's called Linclust. It's a very fast clustering method for proteins, but you can also use it for nucleotides now. So just to explain what clustering is in a sequence space is you have a set of sequences here. You can see them on the left side. And the clustering tries to find centroid sequences. So these are these green sequences here that capture a lot of these other sequences based on some clustering criteria, for example, sequence identity and so on. So you can imagine that all of these dots here in this area have the same sequence identity. And what you can use that for is you can use it to reduce redundancy. So you can just remove things that are looking similar. You can use the MMseqs2 and Linclust workflow actually to also build protein families. You can cluster things that are also really far away at like 30% sequence identity or so. And once you have your protein families, you can build consensus sequences and so on. So clustering can be really powerful and help you to do your analysis, sometimes even faster by just removing the redundancy. And Linclust scales linearly with the input size, while most other clustering software, or I think by now still all the other clustering software scales quadratically with the input size. So what you see in this plot here on the x-axis is the amount of sequences in millions that we clustered. And on the y-axis is the runtime in minutes. and we used MMseqs1, CD-HIT, Uclust, and then the Linclust workflow with MMseqs2 in like multiple flavors. And also we used MASH. And we can see that all of these tools have some quadratic tendency. However, Linclust just scales linear here. So we did it at like 90%, 70, and 50% sequence identity. So there are like multiple parameters we have used for clustering it. It's also very fast for like 50% sequence identity. So it still stays linear. So the question is now, how do we achieve that? Like, how do we make our method scale linear and not quadratic? So the idea here is actually fairly simple, I think. So you have these black dots are sequences, and these sequences have k-mers, right? And what we do with Linclust is we just extract 20 k-mers that are having a lowest hash, but it doesn't really matter if it's lowest hash or not. You can just extract all of the k-mers. The idea would still work. The idea is you're building these k-mer groups, meaning that each of these sequences in this green circle shares the same k-mer, having the same k-mer. Each of these dots here having the same blue k-mer, and so on. And the trick now to become linear in the runtime is that for each k-mer group, you just pick the longest sequence and say the longest sequence in that k-mer group is the centroid of that k-mer group. And this centroid sequence can be shared, so it can be the same sequence also for the blue and green dot here. You see that the red dot here is the longest sequence. It covers the red, green, and blue k-mer group. So once you have done that, what you do now is you just align every sequence that is in the k-mer group against the representative sequence, just to check if it fulfills your clustering criteria, like sequence identity, coverage, whatever you want, right? And this is right. And this was in general, by picking the longest, we avoid doing this. Before, normally clustering methods have to look at all of these combinations and then pick an centroid for the clustering. We make the approximation in general and say that the longest one is it, and therefore we don't have to do all of these alignments and scale linear in runtime. And yeah, once we have established our connections, we then just cluster them. We just have to make some separation between these groups so that sequences are not assigned to multiple clusters. Let's run the Linclust command, actually MMseqs2/Linclust command on these pathogen assembly data. So we enforce some kind of coverage here. Currently, I guess we do not even set a sequence identity. You can also set the sequence identity. We set the coverage mode 1. I will go over the coverages modes for afterwards, because I see often users of MMseqs making mistakes when they set the coverage modes for the clustering. Okay, it is clustered. Let's explain the coverage modes here. So we have the coverage mode 0, 1, and 2, and 3. Normally, we only use 0 and 1. I think this is the mostly widely used one. 0 is something if you want both sequences to be nearly identical. So meaning that overlap should be in bidirectional overlap. So the alignment should span 80% of the query, or like centroid, and 80% of the target. So you're assuring that the length is kind of similar between the sequences. And for mode 1, you want that the shorter sequence of both of them is covered at least at that coverage threshold. And for mode 2, it's similar to 1, but vice versa. And mode 3 is very specific, which we have implemented for the UniRef, which is currently using MMseqs to build the UniRef databases. Okay, once you're done, you see multiple output files, because it was an easy workflow. It again tries to generate as much things for you that you don't have to generate them afterwards and more difficult. It generates an all-seq FASTA file, which should contain all of the sequences assigned to the clusters. It should have a cluster.tsv file, which is just an identifier mapping like telling you which identifiers are in which clusters. And then there are the dereplicated sequences in the rep-seqs. These are the representative sequence FASTA files. So we are, so we can have a look at, for example, these formats. The rep-seq format here is just a FASTA file with the centroids, and then the assembly cluster file is an assignment of sequences, for example, meaning that here sequence 0 is in cluster 0 and sequence 1 is in cluster 1. So they are both not clusters, those are just singletons. But here, for example, would be a cluster. Sequence 3 is in cluster 3, and sequence 27 is also in cluster 3. Normally, these are the FASTA identifiers. Since PLASS has just generated 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, they are just 1, 2, 3, 4, 5, 6, 7, and so on. But if you use UniProt, that will be UniProt identifier or whatever whatever identifier you are using. So the question is now how many sequence remains now? You see that a lot of these sequences got cut out, so we only end up with 17 sequences left. There was a lot of redundancy, as mentioned. The PLASS assembler doesn't care about redundancy; it produces a lot, so it's always favorable to run the clustering afterwards. Okay, we can now download that protein, that polyprotein from the UniProt side, pipe it into MMseqs Easy Search to compare that polyprotein with what we have assembled. So it's a bit of an advanced command here. So it really just downloads the file, but it just downloads the file, it performs the search, and then it uses some format output for the MMseqs. That MMseqs produces some HTML rendered output. Makes it maybe somehow easier to look at this kind of thing. Okay, the command is done. And now let's have a look at that result output. And we can see now that here is the contig. This is the protein, the polyprotein. And each of these hits is something that was assembled by the assembler. So you can see that only by looking where these hits are, there's quite a lot of sparsity in that assembly. So it just hits like small pieces of that polyprotein. You can scroll down and you can look at the alignments here and see how well the things align and what kind of sequence identity they have. And like use that to explore the data. But yeah, for a first look, it looks like we don't capture a lot of that pathogen. But the good thing is that hits are like spread out all over, right? So they are not just like locally at one point, so they are somehow all over the polyprotein. If you map back the reads, you're ending up in quite noisy signals. So what you can see here on the lower part is the sequences that are assembled by the PLASS-HYBRID, and then you can see the sequences that are assembled by megahit. So you can see that many of the sequences assembled by megahit are also the same with the PLASS-HYBRID. So both assemblers do fairly similar in their performance. Here the PLASS-HYBRID could capture some more pieces that the megahit does not have. But overall it looks very similar. But the way how the sample was treated really caused some biases in the data. I haven't really explained how the treatment of the sample was. They use some kind of host depletion and which can cause biases in your data. And yeah, you see that the coverages do not look as the way how we would expect it. We would not expect like these huge bar plots here. Would want to see the reads kind of being distributed all over. One more thing, the last thing I'm talking about is the MMseqs2 web server. It's at search.mmseqs.com. You can set it up yourself. It's an easy to install server that you can use certain databases you want to search against. Here I have already done a search yesterday with all of the contigs that were assembled by the assembler. So I've just linked that result here directly, so I'm not performing the search. And what that does now is that the server is showing you all the queries here on the left side, which are the contacts. So currently we look at the zeros contact, and you can see the hits that it have to the PDB, like mean the structural database. There is some viral orthology database. There's a UniProt hit here as well, and the PFAM annotation. So it performs all of these searches against multiple databases for you at once. So you don't have to really go through all of these databases by hand. You just like put in your sequences and you see, oh, there's a CDD. So there's some domain. There's often the CDDs and the PFAMs are overlapped because they should be a subset of each other. You see the Uniclust sequences and PDB sequences and so on. And yeah, you can click through that. And here, yeah, there's some viral orthology group here. So might be useful to like just explore your context in a fast way. And yeah, again, that interface also shows you the alignment. So you can click on this, and then you will see the pairwise alignment between these hits. Okay, so what happened? First of all, the Powassan virus that we talked about here was confirmed to be the causal agent of this patient. They could find it using these NGS methods. They have used Kraken for their analysis, not MMseqs2. Kraken has matched 270 reads, I think, to that. MMseqs has matched 1,200 reads to the Powassan virus, meaning that there is a bit more sensitivity using the protein level matching over the nucleotide matching. Unfortunately, the patient did not survive it. It's still in quite interesting case, but yeah, a sad ending. So I'm sorry to ruin the mood with this. So you can look at that study. I have linked it here. Also, all of this comment and everything I have used here, I will share, and please feel free to reproduce that study and work with the tools. Yeah, here again the references. And I want to thank my lab at the Seoul National University, and also the Söding lab that is heavily working on MMseqs and PLASS. And I want to thank you for listening.